now we start in module 3, which is including GWAS and using epigenetics to understand GWAS studies, and also single-cell RNA-seq and single-cell TAC-seq. So first is GWAS and SNPs. If you compare between two individuals, vast majority, 99.9% of their genome are identical. There are some regions that are different. You and I, we have 0.00100, yeah, it's very tiny percent of regions that are different. And there are some that are kind of common. For example, there are some regions with a single nucleotide in some populations, say the Caucasian population. You might see a G, but in an Asian population, you might see a T, or African population, you see a T. And so these appear in significant populations. So, you probably want this to be over 1%. These are called, so first of all, the locations where these things happen are called locus or loci if you have multiple locations. And the variations in this location are called alleles. And so in this particular locus, we have two alleles, G or T. In order to call something as polymorphism, the minor allele frequency needs to be over 1% of all the population. Otherwise, this region is called a rare variant and not really a polymorphism. And if you compare between two individuals, the most frequent difference is the single nucleotide like this. Occasionally, you might have small insertion deletion of, say, a single a few nucleotides. And these are much rarer. There are also copy number differences. Some regions, this person have 50 copy, another person have 20 copy, but those are rare. 90% of the human genetic variation happen in this single nucleotide differences. And sometimes adjacent these SNPs can be linked. And so for example, if at one locus, I have two alleles, say CG. In another locus nearby, I have also two alleles, say GT. And in a third locus, I have another two alleles, say AG. And if you look at so many many people in different populations, you might notice that whenever you have a C in this position, these two are usually together, and these two are appearing more often than expected, the two alleles. Whereas the ones that are further away, because of recombination in the population, the most, the really furthest away is that, the C locus is in a different chromosome as your AB. And then they are really quite independent of each other. Whereas if the C is on the same chromosome as AB, but kind of further away, you will see that they are sometimes still they are linked. And so in order to see that, these are called the linkage disequilibrium or Hardy-Weinberg disequilibrium. For example, very simple, we look at, if you see a population with AA types, this is just looking at a single locus. Say this position can be AA, or the small a means that it's the minor allele. Minor allele means it's appearing in less than 50% or the minority of your populations. So it could be that the major allele is a G and the minor allele is a T. And so you're asking what's the T percentage and the G percentage, and also the mix, because each person have two chromosomes. And so if overall in the population, you have A at, in this case, 60%. So the major allele 60%, the minor allele 40%. And this is what you actually expect to happen, right? Because you assume everybody random chance of getting this from the mom and dad copy. And so if in the population, 60% of major allele then the likelihood they are having two major alleles in this location is 36%, and two minor alleles is 16%, and the mix is 25% of each. And now if we look at two adjacent loci, and each have allele, big allele, minor allele, major allele, minor allele, you could see that by chance we would expect this mix to be AB as 36% and the small AB as 16%, but in reality you might actually see a different percentage. And so the big A and the big B happen a lot more often together, and small a, small b also happen a lot more frequently than expected, whereas the small a, big B or small b, big A are happening a lot less. That means that this A and the B allele is likely to be linked to each other. The reason for that is because these two are nearby on the same chromosome, and so during recombination they always go together. Whereas something that's on a different chromosome or really far away on the same chromosome, you probably will get more of this equilibrium kind of behavior. And so linkage disequilibrium means alleles occurring together more often than expected, meaning that they are physically close to the DNA. And if you look at how across the genome in regions that are linked, you see these triangles. This is, which means that you see very strong LD with each other within this region, but not strong LD across each other. This is almost like a TAD domain, right? You are seeing these triangles. I suspect you can also use the hidden Markov model to identify those LD blocks, right? We can decide, okay, these regions are one region, there's another LD block, there's another LD block. And usually the block boundaries reflect the recombination hotspot. It means that during meiosis and the DNA recombination, usually there is a DNA breakpoint, and this chunk can be exchanged for the other copy of the chromosome from exchange between the mother and father copy, and so the breakpoint here is usually a haplotype boundary, and because a lot of the SNPs within the same haplotype blocks are linked to each other, we'll show you later on association studies for looking at whether particular SNP is associated with the disease. If you use every individual SNP to do the association, there are too many regions or too many SNPs to do the test, and you usually have to use a very stringent p-value criteria to give you enough hits, or to give you hits with a confident enough false discovery rate. And so one way to increase the statistical power is to use all the SNPs within one haplotype block as if this is only one SNP or one chunk of SNP together. And so this way the genome is partitioned into haplotype chunks. You'll reduce your multiple hypothesis testing significantly and increase the statistical power. And so why do we care about SNPs? This is because certain diseases are known to influence populations differently. For example, you probably heard that Angelina Jolie, because of a particular SNP on her body, she is more likely to have breast cancer. And so she decided in a fairly early age to have double mastectomy to remove her breast so that can reduce her likelihood of developing a disease. And there's another study was published in Science several years ago. This is Mike Snyder, who is the chair of the genetics department at Stanford. He kind of did his SNP profiling, and based on the knowledge at the time, he was predicted as more likely to have diabetes. And around the same time of the study, he actually saw some early warnings that early onset of diabetes signs. And so he started changing his lifestyle in order, like eating healthy, exercising, and just change his lifestyle in order to cope with the type 2 diabetes. So I think he's actually now still healthy, not having diabetes. And so the SNP profiling can help him, make those lifestyle decisions. SNP also have very, very important effect on patient response to specific drugs. I apologize, I don't have the slides there now, but depending on the specific SNPs, some patients might respond better to a drug than others, and this has been documented before. And so in order to do these type of association studies, there are two different major types. One is called family-based association studies. And so for this, you usually get the mom and dad and you profile them, and you also profile an affected child. And sometimes you also profile one affected child and one unaffected child from the same parents. And so basically for this, you can, early days, people use SNP arrays to use microarrays to profile the different SNPs at each allele. And recently, people are also doing whole exome sequencing or even whole genome sequencing. Now you really read out the genome wide. And then for the polymorphic regions, or just look at the SNPs locations, you can ask, what is the allele at the father? What's the allele at the mother? And what's the allele in the child? And so this is one a way of doing the family association study, which is to look at the affected child. We know, so this is some traits, say the father and mother have this and this allele, and the child, let's say diabetes. We know this child also have diabetes and, like their parents. And so after this, we look at, the one family, another family, and third family, and so on. And we look at, in the child, we know this child has the disease, or some phenotype, say blonde hair, or some blue eyes or something. We call it affected child. And we ask, what is the allele that's being transmitted from the parent? So you will see that some alleles, we have no information, then we don't consider it. For example, in this parent, there's no selection of which allele get transmitted to the child, because the parent is homozygous. And so there's no there's no choice. You always transmit the big A to the child. Whereas in the second parent, there is a difference. Because we know the child is too big A, then we know from this parent, it's the big A that's being transmitted to the child. So the important information is on the heterozygous parent allele that's transmitted to the child. So that's only one A in here. In the second in the second case, both parents actually are important. And so we know there is some choice made. First, a big A is being transmitted. Second, a small A is being transmitted in here. And for the third family, this parent has no information, because regardless, they're going to contribute a small A. But for the first parent, we know, again, the major allele is being transmitted. For the fourth family, there's no information. The fifth family, we know that the big A is being transmitted from the dad, and the big A is being transmitted from the mom, and so on. And so at the end, we say, what do we expect? Well, first of all, we look at how often the big A is being transmitted, and how often the small A is being transmitted in this case. In our situation, these two are 0. And then to do a statistical test, basically you can imagine if the affected child, you always see in this case, the big A seem to be transmitting a lot more than you expected. We want to kind of do a statistical test to ask, you can see here, only the B and C are important. There's ADs are all 0, so they don't contribute information. We use this TDT, or the transmission disequilibrium test, to ask, B plus C is kind of all the important transmitted one. The B minus C squared is kind of how unexpected they are, the difference between the B and C. If there is no difference for this allele, you can see that B and C might be quite even, then there's no information. Whereas if the you see in this case, a big A seem to be transmitted a lot more often than a small a. Then the B minus C is a significant difference. And using a chi-square test, you will see that, oh, that it's a lot more different. And so in this case, it's the big A minus small a squared divided by the sum of the two. And you check the chi-square table, and you will see that it's a very significant difference. You can also, in this case, compare the allele frequency between the affected child and unaffected child from the same family. And then also look at how much the big A allele versus the minus A allele are being transmitted, but it's a slightly different equation, but it's similar kind of idea. Are we seeing the one allele transmitting to the affected child a lot more than it transmit to the unaffected child? Those kind of these type of tests are quite powerful, but to collect the family sometimes is tricky. Because sometimes when you find the child with the disease, the parent has already, especially the disease that happen with age, it could be that their parents are deceased, so you can't really get the DNA information anymore. And so in addition to doing the family-based association, the more common approach is to do the case control studies, just population-wide studies, such as you just get, say, most of the people, you take a patient from 40 to 60 60 years of age, and then you take all the people who haven't got diabetes at this age and versus people who have diabetes at this age, 40 to 60. And then you look at, again, their SNPs. And in each case you ask, how often, this is at one locus, you ask, how many times do we see GG? How many times do we see GT? How many times do we see TG, right? And you ask, what's the number of cases that you see in this case? But here the transmission is different, or the actual allele counting is different. Because if you have a GG, there are two Gs at in this patient, but only one G in this heterozygous case. So in the cases, we actually see two Gs, the R0 times 2, and the R1 is just 1, right? And then you also ask in the controls, how many cases do we see as GG? And that is multiplied by 2. And how many times do we see a GT heterozygous case? And that's only 1 G. And then T, you calculate, in the TT, it's counted twice in this case. And so then at the end, you ask how many total times the G allele appears, the T allele appears. And then you ask, what is the expected alleles? So because we know in the total population, we see the G alleles appearing out of the total, basically it's the G divided by the G plus T, the expected of G in the cases and the expected G in the controls. You can ask in the actual observation from the case and control, how different is my expected from the observed? And so if you have, observed allele as this and expected allele like this, and you can ask, how often do we see this? You can again use the chi-square, which is the expected subtracted by the observed squared divided by the expected. And again, this is kind of a chi-square test. And for this, let me ask you guys, we'll get a p-value. Do we need to worry about multiple hypothesis testing for testing the SNPs? You do, because you have to do this chi-square test across every SNP in the genome. And so you will be doing millions of chi-square tests in here. And that's why sometimes in order to really see that, oh, this particular SNP is associated with a disease or a phenotype, you have to have a huge population to give you that statistical power. Otherwise, in order to get you a reasonable FDR. And so one solution is to have a huge population. The other choice is to do this test at haplotype blocks, because we know that SNPs within the same haplotypes are usually linked. They are always together. And so you only need to do by the number of haplotype blocks rather than by the number of SNPs, which will give you significantly fewer tests and stronger, better, you can use kind of a higher p-value to reach the same FDR. Another important issue is odds ratio. And so you can imagine the more people you have, the stronger p-value you have, but the actual disease association might not be very, very strong. So you are asking, you know, the effect size, odds of an event occurring divided by the events not occurring. So if I have this SNP, am I, 80% likely to have the disease, or am I more likely to have the disease? Am I twice as much likely to have the disease, or am I only 1% more likely than other people to get the disease, right? So that's kind of your effect size. And so you're looking at the, like the alleles in G in case and the allele G in the control, the allele in the T in the case and control, and considering all the G's in the same sample and pick at random, the odds ratio, by chance that in this location, I'm gonna, the likelihood that G appears in a case is A divided by C. So the difference between odds and probability is odds is just A and not A, A divided by not A, whereas the probability is A divided by the sum of the two, right? And so, but then here we're looking at the odds ratio, which is the odds that G allele occurs in a case divided by the odds that T appears in this case. And so this odds is here, the G appears in here is A divided by C, and the odds of T appearing at this case is B divided by D, and the odds ratio is the ratio of these two, and that will give you an AD divided by BC. And so if you have a very big odds ratio, that indicate that if you have you have a very big chance of getting the disease, or a much higher chance of getting the disease. Whereas if the odds ratio is pretty small or close to 1, that means you are only slightly more likely to get the disease if you have this SNP. So you can see in this situation, in the C and the G allele at the same location, the chi-square test is giving you a really tiny p-value. It's because we have a pretty big population here, 2,000 people roughly 2,000 people to do the association. But the allele odds ratio is only 1.38, which means you're like 38% more likely to get the disease, or 1.38 compared to people without that allele. You're only 1.38 more likely to, or 38% likely to get that disease. And so if odds ratio is equal to 1, there's no association. And usually those p-values are not significant. If the odds ratio is over 1, allele C is increasing your disease risk. If it's below 1, it's allele C is decreasing your disease risk in this case. And so you can see here, if the odds ratio is high, you might need fewer samples to reach the same p-value. But sometimes you can have a huge population that give you a decent p-value, but the odds ratio is only slightly different from 1. That means that it have only a weak association with the disease. Okay, so again, you use the FDR to adjust for your p-value. So that's kind of a general overview of GWAS studies or association studies. Over the last decade, we see an increasing rate of GWAS studies. This is because people really want to see, based on my genetic information, can I predict a disease? How likely am I going to have a disease? How also they are interested in whether there's association with the patient's response to drugs. And this is facilitated by the drastic reduction of genome sequencing cost in early days by the microarray cost. And so you can see the number of publications in the GWAS studies are really increasing. In the early days, when the cost is high and people didn't have enough data yet, they also it's kind of collecting sample cohorts is very time consuming. You take decades to collect enough patients for some case control studies. And so in the early days, they were looking at cohorts that have only a few hundred patients. And so sometimes they might find a SNP that give you some association. And if they repeat the same study, again, using another scientist do the same study, they collect another cohort of patients with a few hundred patients, they do the GWAS, they see some SNPs, and there could be very low overlap between the two studies. And this is mostly because in the early days, there were not enough patients. Most of these GWAS individual association have very weak effect to the disease. And and because we're doing so many multiple hypothesis testing, you actually hit on things that are not really real. And if you change a cohort, you'll land in some other regions. And so you can see here, the number of GWAS hits is directly associated with your discovery cohort size. Early days, it could be a few hundred or a few thousand, and then to 20,000 and even 200,000. And now with more and more people doing genome sequencing, you might even have these huge cohorts that have close to 1 million individuals to look at association. The nice thing about this is, once you sequence somebody's DNA, the information is always there, and all you need to decide is which phenotype do I want to look at. You can see here, this could be a height, body mass index. This QT interval is heart QT interval. Cholesterol level, you can look at hair color, eye color. You can look at disease, other association. You can look at their IQ. Yeah, it's just many like skin color and things like that you can look at. So in these type of GWAS studies, to make sure that you are getting the robust result, having bigger cohort is very, very important. Another important thing is to make sure that you don't have population stratification. So for example, if I want to look for SNPs that can predict longevity, I collect people who can live over 100, centenarians who are over 100 years old versus people who died before they reach 100 years old. And, of course, centenarians are hard to find, but you collect those and you do the GWAS study, and you might find, it turns out, hidden things you don't know is the centenarians are enriched in, say, this Japanese population. Whereas the people who died otherwise younger are kind of a random mix. And so because of that, when you are looking at the SNP difference, what you might see is that, oh, the Asian SNPs are all showing up because it has nothing to do with longevity. It just happened that in your cohort, the longevity cohort have more people from Asian or from Japanese. And so in this case, in your case, you have an overwhelming population, whereas your control is different. And so what happens is that when you do the p-value calculation of different SNPs, remember we are doing this for millions of chi-square tests, we can look at the p-value of those chi-square tests and then do a QQ plot to compare the expected p-value and the observed p-value. If you don't have population stratification, you would say that, okay, most of these SNPs are not significant. Then suddenly we have some SNPs that are significant these are real. Whereas in other cases, if you see kind of a very early diversion from your expected like this is an indication that your case control populations have this population stratification. There are a lot of SNPs that are significantly different between your case and control. That could be because of racial, ethnic groups that are differently populated in your case and control. And so for that, you have to correct that and kind of only call those ones as your real difference. Another potential, so this is one example. If you also just run the PCA on all the SNPs, you can already see that on this PCA plot, there are people from different populations. And so, correct for the population difference from your case control studies that are arising from the uneven populations in the different cohorts. Another situation is there are some unknown relatives. Maybe you have a distant cousin in this study that you don't know, or in a case control, we don't want family structures. And so you really want unrelated family members for the case control study. And so there are also something called IBD, which kind of give you the chance that these two individuals are from the same family. And so if on the IBD test, you see, you see a lot of people from the same family, you might want to remove the sibling or the cousins from the study. You really want a representative member from unrelated family in your study. And so by this time, for example, this is a figure in 2019. In NCBI, there is a GWAS catalog. And they continue to collect published GWAS studies and get their SNPs from them to see, this is on chromosome 1, 2, 3, 4. What are the current discovery of SNPs that are associated with different diseases? And so there are like many of those summary statistics files that tell you this GWAS study identified the following SNPs as associated with that disease. And another is a different collection of SNPs association with disease. And based on our lecture in the last Hi-C interaction lecture, Dr. Feng Yue mentioned that a lot of these GWAS SNPs, people found that only a very small set of those are actually in coding regions. Because coding region is only 2% of the genome, a lot of these GWAS-associated SNPs lands in non-coding regions of the genome. And interestingly, a lot of them are enriched in DNA's hypersensitivity peaks. Or there could be some that are not in the peaks, but in perfect LD with those peaks. This is because sometimes they didn't do genome-wide association using genome sequencing. They just use SNP array. They typed one SNP, but they don't have the SNP in the DNA's peak region. But these two are perfectly linked, so you are suspecting that the real effect is on the DNA's hypersensitivity region. And only a quarter of them are not in LD, which suggests that a lot of these SNPs associated with disease are related to a transcription factor that binds to a region which influence the nearby gene expression. This nearby could mean 50 kb or 200 kb or something like quite a distal region that influence the expression of that nearby gene, which then have an effect on the phenotype. And so understanding epigenetics now become very, very important to interpreting these GWAS SNPs. It's like, there's a SNP in here, it's associated with a disease, what's happening? We want to see what transcription factor binds that enhancers and what kind of nearby genes are linked, and then we can understand why that's related to the phenotype or the disease. Another type of study that's really useful is called eQTL analysis, and a national project, a consortium project called GTEx is really, really quite exciting. So first, they look at 1,000 individuals. These are not disease or no specific disease. It's just average people who have died from a lot of them could be unnatural causes or whatever. And so they donated their body to the project, and they take different tissues. In this case, brain, heart, like many different tissues, 50 different tissues from these individuals. And they did both the SNP typing of, this is using SNP arrays to look at their SNPs across the genome, and also to do the expression of those 54 tissues across individuals. And so in here, because we are not interested in disease per se, the trait in here is no longer whether this person have blue eyes or black hair, or whether they have a fair skin, or whether they have high body mass index. Instead, we use expression data as the phenotype. You say, well, it's 1,000 people's liver, but for this gene, it's expressed at a much, much higher level in some population, but lower in another population. What is the difference? And so the features are on the SNPs. And we say, oh, it turns out if they have, in this case, for example, we say if they have these SNPs, the expression is much, much higher in, say, liver. But if they have another SNP, the expression of the nearby gene is happening much lower in liver. And so you can establish this association from something called expression quantitative trait loci. So quantitative trait is, height is more quantitative. It's not a black-white 1-2. It could be just the expression level. And the trait in this case is not really eye color, physical trait. It could just be an expression. And we are trying to look for a particular SNP locus that's associated. The SNP in this locus is associated with a quantitative gene expression level. And using this type of eQTL analysis, again, a lot of people find that there are SNPs in nearby regions that are linked to gene expression differences. And a lot of those are also in the ataxic or DNase peaks, which means that gene transcription factor binding in the distal regions might influence this gene expression and eventually influence real phenotype or trait. Yeah, so that's kind of the summary. In the genome, there are SNPs. They are often linked if they are in proximity on the chromosome. And there are also these haplotype blocks where a lot of SNPs are linked to each other. And in order to really do the genome-wide association studies, you can collect big families with members associated, are known to be associated with the disease, to look at the allele that's transmitted, which made the difference. Or you can just do case control studies, normal versus disease, and check every allele to see whether there is any difference between the allele frequency in the two populations. And in order to increase the statistical power of your analysis, you can look at haplotype association rather than individual SNP association. You could increase your patient sample population. Bigger cohort, the better power. And also, you need to make sure to remove the populations, eliminate the population stratification, and remove the too close family members. This is in the case control studies. And also, we can use the eQTL to identify SNPs associated with differential expression, and also the GWAS SNPs are also linked to some trait. And a lot of these eQTL sites and the GWAS SNPs are in the non-coding regions. And then we need to use the epigenetic data to help us understand what's happening. Okay, so that's all for today.